Hello. So we studied the two methods of transport or transportation across the cell membrane. What are they? One is diffusion and the other one is osmosis. So examples for this diffusion and osmosis. The same examples that we discussed here. For diffusion, we discussed about the transport of carbon dioxide and oxygen across the cell membrane. Okay. Then the osmosis. Osmosis, what is the difference? Here one semi-permeable membrane is needed. So a semi-permeable membrane, usually we are using the membrane called cellophane. Cellophane is one of the semi-permeable membrane that we are using in the lab activities and others. Then another semi-permeable membrane that I will say to you, that is the egg membrane. Egg membrane means not the shell of the egg. After the boiling of this, or in the case of a boiled egg, just after the removal or just below the shell, when we are removing the shell, we can see just below the shell there is a white colored membrane. This is the semi-permeable, one of the semi-permeable membrane. Boiled egg shell white colored membrane. Okay, that is the semi-permeable membrane. And our cell membrane, the living organism's body is made up of thousands and billions of cells are there. All the cell membrane is also made up of semi-permeable membrane. So for osmosis, what we can say the example, that is the swelling of the raisins when we are keeping it in the water. And there are two types of osmosis. One is the endosmosis and the other one is exosmosis. Endosmosis and exosmosis. If the raisins, the example that we can take the example that we discussed now. What is it? The raisins. Okay. So it will become cells up. That is an example of endosmosis. That means the moment the water molecules, they are entering to inside of the cell. Water molecules, they are entering to inside of the cell. That is why the cells become cells up. Then what is the condition of this exosmosis? For example, when we are keeping the tender mangoes in salt solution. Okay. What will happen after 1 or 2 months? The mangoes which will become strings. Okay, so this is because of the entry of water molecules from inside to outside. Okay, that is an example of exosmosis. That means the water molecules, they are coming out of the cells in the portable. Okay, this is a process called exosmosis. So endosmosis and exosmosis. Based on this, we can classify the solutions into three types of solutions. When we are keeping a plant cell or an animal cell in one solution, we don't know what type of solution is that. Okay, we don't know what type of solution is that. Just we kept one plant cell in one solution. So after some time, we can see the cell becomes swells up. The cell becomes swells up. So from this, we can understand that inside the cell, the concentration is very high. Outside the cell, the concentration is very low. What concentration? Water. Water concentration is very low. So what happened? From outside of the cell, water concentration is very high. Inside of the cell, water concentration is very less. So from outside of the cell, the water molecules enters to inside of the cell. This is why the cell becomes swells up. So we can say the concentration of that water or the surrounding solution is comparatively less than that of the cell. This kind of solutions are called hypotonic solution. Hypotonic solution. Hypo means less. Hypo means less. So inside of the cell concentration is very high. Outside of the cell concentration is this is very less. That means water is very high. Water content is very high. So what will happen? Water content from outside enters to inside. So suppose this is a cell. So what will happen? The water molecules may enter to inside. So this surrounding solution, we can say this is a hypotonic solution. One type of solution is that. So in the second type of solution, we just changed the solution by using another media. So replace the solution or keep the cell in another solution. At that time, you know what happened? The cell becomes strings. the cell becomes shrinks. That means outside of the cell the concentration is very low. So inside of the cell water concentration is very high, outside of the cell water concentration is very low. Such kind of solutions are called hypertonic solution. Hypertonic solution. The hypertonic solution means the cell with the solution with the higher concentration. Okay, that means water concentration is less. Amount of water is less, but the solute concentration is high. Then again we replace the cell or we replace the solution by using another one. At that time there is no change for the cell. Cell churungiyadilla, there will be no swelling up and there will be no shrinking. That means there is no change in the concentration. Inside of the cell and outside of the cell shows the same concentration. Okay, ഒരേപോലെ ഇൻസൈഡ് ദ സെല്ലിലും അതുപോലെ സറൗണ്ടിങ് മീഡിയവും ഒരേ കോൺസെൻട്രേഷൻ ആണ്. സച്ച് കൈൻഡ് ഓഫ് this cells or solutions are called isotonic solutions. Isotonic solution. So three types of solutions we discussed: hypertonic, hypotonic and isotonic. Hypo means lower. So the cell solution with lower concentration than that of the cell, hypotonic. Then hyper means higher concentration. The solutions with a higher solute concentration than that of the cell. Then iso means same. The solution with the same solute concentration like that of the cell. So these are three kinds of solution. So this is the method of, in this way the materials such as solid, liquid, gas, whatever it may be, they are transporting across the cell membrane. So that's all about this. Thank you.